Hello everyone, welcome to our channel Ashwatha Edu-hub, and today we are going to continue the part of photosynthesis in plants. So as we know that last class we have studied where photosynthesis occur and what is the mechanism of photosynthesis, and we found out that photosynthesis occurs in two phases, one light reaction and the other is known as dark reaction. So we studied a bit about that light reaction. So we are going to continue with the light reaction. So in light reaction, what happens? Energy from sun will be converted into ATP and NADPH2. For that, what is required? Pigments. And pigments, they are not scattered. They are actually found as a unit. So pigments, they are found as a unit on the membrane of thylakoid, and that unit is called as photosynthetic unit. It is also called as what? Quantasome. Clear? Now, what does this photosynthetic unit consist of? It consists of chlorophyll and carotenoids. So when we talk about chlorophyll structurally it consists of a porphyrin head and a phytol tail. This is the porphyrin head. Clear? This porphyrin head consists of four pyrrole rings. Pyrrole ring number 1, 2, 3 and 4. The four pyrrole ring together they constitutes the porphyrin head, and the phytol tail is attached to the pyrrole ring number 3 of the porphyrin head. So this phytol tail, what happens? The tail it attaches, it helps in anchoring the molecule. ধরে রাখার সহায় করে. Clear? Now chlorophyll A and chlorophyll B, we know they are the main pigments of photosynthesis. We find that there is only one difference between chlorophyll A and chlorophyll B. In case of chlorophyll A, it possesses a functional group which is a methyl group. In case of chlorophyll B, it possesses a functional group which is an aldehyde. Rest of the molecule, it is exactly same. Only the functional group differs. Clear? So here I'll write the empirical formula of chlorophyll A. You try to find out the formula for chlorophyll B. So chlorophyll A, it has an empirical formula of C55, H72, and 4MgO5. So it's very easy to remember actually. If you look into the pyrrole ring, 1, 2, 3, 4, all the pyrrole rings, they have one nitrogen. This is the one nitrogen, second nitrogen, third nitrogen, and fourth nitrogen. Clear? And 4. And all the 4 nitrogens, they are attached to a core of magnesium. So how many magnesium? 1. This is definitely common. Now your homework is to find out the formula for chlorophyll B. The difference is what? In case of chlorophyll A, the group is what? Methyl group. In case of chlorophyll B, it is aldehyde. So try to write it down and let me know in the comment section. Clear? Now, when we talk about a unit, definitely there will be like a number of pigments present there. So we find that one photosynthetic unit, it consists of approximately 230 chlorophyll molecules, and in that unit we find that chlorophyll A is abundant, more than chlorophyll B. So this is how a unit will look like, okay? They will have chlorophyll A molecules, B molecules, it will have carotenoids, they will be all forming a unit, clear? And of all the molecules, one of the chlorophyll A molecule, it acts as a reaction center. This is the primary chlorophyll A molecule, which will directly participate in production of ATP and NADPH2. How, we will know later. Everyone participates definitely, but this one is the main or the key one. So that is called as what? Reaction center. And it is always a chlorophyll A molecule. Clear? Now apart from this A molecule, yak bad diya, a chlorophyll A molecule jeto ase the main reaction center, leaving that rest of all the molecules, other chlorophyll A molecules, B molecules, carotenoids, they all form what? Antenna molecule or accessory pigments, or it is also called as light harvesting complex. Why? Because other molecules, they funnel or they harvest the light to reaction center chlorophyll A molecule. So that is why it is called as light harvesting complex. Clear? Now if you remember, this is the thylakoid. This is the membrane of the thylakoid. Inside of the thylakoid is called as what? Lumen. Okay. Now this photosynthetic unit, it is present on the membrane of the thylakoid, clear, as a unit. So I hope this is clear up till now. Now another homework for you. Chlorophyll is soluble in what kind of solvents? Clear? So that's a homework for you. As I have told you earlier that chlorophyll, carotenoids and biliverdins or phycobilins, they are soluble in different kind of solvents. So today your homework is to find out in what kind of solvent is chlorophyll molecule soluble. Clear? Now, in the photosynthetic unit, apart from chlorophylls, we will also have carotenoids. Now, these carotenoids, they are also called as what? They are also called as lipochromes. Why? Because they are soluble in lipids. That is why their other name is lipochromes. Now, what is their function? So, one function we know that they will funnel the energy to the main reaction center, chlorophyll A molecule. Apart from that, they also helps in prevention of photooxidation, because when a high energy light will fall on the pigments, on the chlorophyll apparatus, there can be photooxidation, you know. So, they will also prevent that. So, that is the role of carotenoid pigments. Clear? Now, we have to know a very important thing is we know that pigments, they can absorb the visible spectra of light, which ranges from 400 to 700 nanometer. Now chlorophyll A, chlorophyll B, carotenoids, they will definitely have what different, you know, absorption peaks. So let us see that now. Clear? See it graphically. Let us put the wavelength of light on one axis. Okay, this is the wavelength of light on one axis. Suppose on axis x we have wavelength of light. What will be the range? 400 to 700 nanometer. Yeah, this is the range. And one end we will put the absorption. So jigita wavelength of light dot peak bilak dangor hobo tat ami ki gom pam je etu light besike absorb korise. So let us first see for chlorophyll A. So it is somewhat like this, you know. So this suggests what? If you see here, if I write here, somewhat like this, if you see, we find the peaks near what? Blue region and the red region. So this is for chlorophyll A and this is for chlorophyll B. And we find that both the pigments, they have absorption peaks near which wavelength of light? Blue and red. So from this we come to know that although we say that pigments, they absorb the visible spectra of light, two wavelength of light, that is blue color and red color, these two are the prominent one or the significant one. If you look at near like green color, you know, you will find that in the green the peaks are nil, you know. It suggests that green color is the most reflected light, while blue and red color of light, they are the more absorbed light. That is why we find higher peaks in blue and red region of the spectra. Clear? Your homework is to find out the absorption peak for carotenoid molecules. Clear? So if we plot wavelength of light on one axis and on another axis we put rate of photosynthesis, what will you find? We find that there is a correlation between the absorption peaks of the molecules as well as the peaks of rate of photosynthesis. So rate of photosynthesis is somewhat like this, you know. So we find that rate of photosynthesis is somewhere falling on the same wavelength of light where we found the absorption peaks of chlorophyll molecules. Clear? So the peaks are near blue region and near the red region. So this is actually called as the action spectra of photosynthesis. It suggests that the rate of photosynthesis is highest when we give what? The blue and the red region of photosynthesis. So there may be questions in NEET, like they may ask you, what are the wavelength of light in which photosynthesis is most efficient? The answer will be blue and red region. Okay. Now we come to another important thing which is called as red drop. So there is a scientist called as Emerson et al. Et al. Means his co-workers. So they were working on monochromatic light. They were working on green algae. So what did they do? They took green algae and illuminated with monochromatic light. Monochrome means single color. So when they were experimenting with red light, they found that when they put red light beyond 700 nanometer, 700 যেতিয়া বেশি wavelength of light they put, imagine that this is a green algae. So this is a green algae and they were illuminating it, okay, with monochromatic light. So when the wavelength of light it was more than 700 nanometer, they found that suddenly there was drop in the rate of photosynthesis. And 700 is the wavelength of what? Red color. So they gave the name what? Red drop. So they suggested that if the wavelength of light is higher or greater than 700 nanometer, there will be fall in rate of photosynthesis. That is only called as what? Red drop. Clear? Later on, Emerson did another experiment. Clear? And he found an interesting phenomena which we now call it as immersion enhancement effect. So what is the meaning of enhance? Enhance means to increase. Clear? So he found that this time what did he do? He simultaneously used shorter wavelength of light. He took 680 nanometer of wavelength of light. He also took 700 nanometer of light. Simultaneously, shorter wavelength and greater wavelength simultaneously. একেলগে লৈছে. Now both the light were used to illuminate the green algae. Clear? Now what he found, now he found out that when you use both shorter and greater wavelength of light together, there was increase in the rate of photosynthesis. And he called it as what? Enhancement effect. In a simple language, if I say, if you use only the greater wavelength of light, what happens? Photosynthesis decreases. That is called as red drop. And together, if you use shorter and greater wavelength of light, rate of photosynthesis increases. That is called as enhancement effect. And after his name, we call it as immersion enhancement effect. Now, this experiment was very, very important in the history of photosynthesis. Why? It suggested that there might be presence of what? Two separate or discrete pigment system or photosystem. Just now I was talking about the photosynthetic unit. That is also called as what? It is also called as photosystem or pigment system. Okay? Clear? And immersion enhancement effect, it led to the discovery of two discrete photosystem or pigment system. So in one pigment system, what will happen? The reaction center chlorophyll A molecule, it will absorb what? It will absorb 680 nanometer wavelength of light. Okay. And the other pigment system or photosystem, here the chlorophyll A reaction center will absorb what? It will absorb the greater wavelength of light, that is 700 nanometer. Okay, now this is called as PS2 and this is called as PS1. Now you might ask me like why the names are altered. Yeah, the this is shorter wavelength, maybe it should have been given PS1 name, and this is 700 nanometer, so it should have been given the name PS2. No, actually what happens, this pigment system was discovered earlier than this, so they proposed the name PS1 for this, and later on they did not change it. Clear? So this question came in previous one of the previous NEET examination. They asked a question like immersion enhancement effect led to the discovery of. They might give you different options. The correct answer is what? It led to the discovery of immersion enhancement effect led to the discovery of to discrete or separate pigment system or photosystem, and they are called as what? PS1 and PS2. Clear? So next class we will continue with another part. I'm giving short videos so that uh, let's first acclimatize ourselves with the topic, and in dark reaction I'll complete in one shot. Clear? I hope till now you are understanding light reaction. See you again then with the other part. Next part we will see how exactly ATP and NADPH2 will be synthesized with the help of these pigment systems. Bye bye.